We've been covering plant senses. So we were looking at bioacoustics, the last one, thinking about how they can hear and produce sound. The last topic on plant senses, I guess, we still haven't covered the idea of vision or sight. And so this is a contentious topic and is probably met with, well, quite a bit of pushback. It's still in its infancy on whether or not, like, how could this be happening? Is it really happening? What I'd like to do is talk you through kind of the progression of how this started to be looked at more seriously. And I haven't had a chance to post, but I have a series of short readings, papers that I want you to read in order so you can see the progression of how people were thinking about this over the years. And I think it's more enriching if you can see the order and the drama unfold. It's like a soap opera. And so, a lovely quote here by Charles Darwin. He was infatuated with orchids, and rightfully so. And he has a nice quote there about their beautiful adaptations. So we need to think a little bit about them first. So need to talk about mimicry, plant mimicry. You might have experienced animal mimicry and have some experience with those ideas, but let's think a little bit about what's happening with plant mimicry. And it does have something to do with vision, so hang with me here. So there's a couple of things to think about. Here we have two examples of two different lovely orchids that are looking the way they do through the process of coevolution with their pollinator. The one on the left is the bee orchid, the one on the right is the wasp orchid. And so this is the idea of coevolution between the orchid and the pollinator, where they are definitely influencing each other's evolution over time. And there is a couple of videos. Let's see if I can get the link up and running. Hang on. Whoops. Hang on just a second. I think it is here. All right, so let's look at the bee orchid. Can I get this to work? And of course, the silly ads. There's no way to like nix out ads, is there, on YouTube? Are we just stuck with them? There's no AdNix app. There's an app for that. AdNix, ad blocker. I don't think it works on YouTube, does it? So annoying. Because I have ad block on my computer. Okay, let's listen. This is Sir David Attenborough. Whoops. You can hear him, but you can't see him. Actual one. All right, let's go. So you can actually see what's going on. Oh, this stupid thing. Go away. All right, I'm going to restart it. This is Sir David Attenborough talking about this orchid.
Do not give their pollinators any reward of any kind. In the warmer parts of Europe lives a whole group that bamboozle their pollinators
Bamboozle.
that they're going to get a really sensational reward, a sexual one. They're little orchids, and their flowers reproduce remarkably closely the signals that enable a male bee or wasp to recognize a female of the same species. Several have blue patches. One is fringed with what looks like fur. A wasp's wings in the right light do flash iridescent blue, and its abdomen is covered with thick brown fur. A female wasp also pumps out an identifying perfume, but the orchid does the same, and the result is irresistible. As the male wasp nuzzles forward in his attempts to mate, he butts the pollinia which stick to him like yellow horns.
That's where the pollen is.
He seems to be well aware that something has happened to him, but there's nothing he can do about it, and he flies off to try his luck elsewhere. Which, of course, is what the orchid requires, because this time he deposits the pollen on another bogus female.
So there, he just pollinated that one. So job done. The plant's like, yep, job's done.
The hairs on many of these orchids run downwards, as though the female is sitting with her head up. But some reproduce her clinging head down, and then the male must land that way if he wants to mate, and he will get the pollen stuck to his rear. This one too seems fully aware that he's got rather more than he bargained for. The orchid's mimicry is so convincing and enticing that sometimes the flower will attract a whole scrum of sex-crazed suitors. Some are trying to get to the orchid and will inadvertently deliver the pollen. Other males, since there seems to be a full house, attempt to mate with one another.
Those are some confused bees. But from the plant's perspective, pretty good. Pretty good process there. All right, there's one other one, another short one I want to show you. Why won't this go away? How annoying.
Now, nectar is made specially for the purpose, and it's costly. And there are some flowers that get away without producing nectar, like this orchid. This is a hammerhead orchid. It mimics a wasp, and a wasp comes and thinks that it's a female wasp and tries to mate with it. There's a very spectacular hinge there that you'll see the use of. There are the pollen sacs, there are the pollinia, and there's the hinge, and there's the fake female. Now look what's happened. The male bees come and been dashed onto the pollen there, going back and forth on that hinge, banging away. And sooner or later, the pollen sacs will come off onto his back. There he is now with his back there, and there are the pollen sacs on his back. And he flies off, and then he'll mate with another flower, and just the same thing will happen. This is a bucket orchid, even more ingenious.
This one's, well,
dropping fluid
we'll watch
into the bucket there.
This isn't really
attractive
mimicry.
fluid. There's a special kind of green bee which is attracted by that fluid. It comes to the flower and it falls into the bucket. There. It's trapped there. The only way it can get out is through a special hole that's provided for it by the orchid. There's the hole. It's the only way out, and the bee has found it. It's forced out through that hole, and on its way out, there is the pollen sac waiting. So the only way through is this little passage. There are the pollen sacs, and it's going to get straight off onto the bee's back on the way out. Now that same bee, when it finally escapes with the pollen sacs on its back, is going to fly off, and it will eventually come to another bucket orchid, and exactly the same procedure will be repeated. It will nearly drown, it will find the hole, and on its way out, the pollen sacs will in this case be scraped off, and they will fertilize the next orchid. So this is deceiving bees, but again, using their wings to carry pollen about.
So that's Richard Dawkins, another famous scientist. Kind of feel sorry for the little bee there, getting nearly drowned and then have to escape through a tiny hole. So the bucket orchid isn't exactly what we're trying to get across today, but it was fun to watch it. But the other one with the wasp on the orchid looking like a wasp but on the hinge joint there. So this, whoops. No, don't play. Don't play. Okay. So mimicry, they often refer to orchids as deceivers or manipulators. You can kind of see why now. And so they're mimicking the look of the pollinator, in this case, the female, even smelling like her, female. All right, now. Extend. "20 clicks later, we're back." Okay. So very interesting coevolutionary process. So this idea is a little bit different than what we're going to get into next, but here's another example of mimicking where this is another orchid and it has these big leaves with spots on them. And when they were trying to figure out, so these are really large leaves with these not so attractive spots on them. And when they were watching to see what would come to pollinate, it was a fly thinking that those spots was a fungal pathogen attacking the plant. And the fly wants that fungus for food. And so the orchid is mimicking the food for the fly, like, hey, I just have all this fungus, come and get it. And in the process, the orchid will get pollinated with the fly. When they looked more closely at the leaves and looked at the trichomes of those leaves, they even look like fungal spores under, the microscope. They could see that the mimicry is that good. And then they tested the volatiles and it mimics the fungal pathogen volatiles. So incredibly sophisticated mimicry going on here for the orchid to attract its pollinator. So again, this is the idea of coevolution between the orchid and its pollinator. And so they often refer to the orchids as, they're deceivers or manipulators. And when you look at orchids, they're hugely successful. It's kind of hard to believe that when you live in this part of the world because you don't see orchids hardly at all around here, but they're really, really abundant. And so 26,000 species and counting. And you have to unfortunately go tropical typically to see them, although we do have lady slippers here, but they're terrestrial ground dwelling orchids. Most orchids are epiphytes. They like to hang out on trees and branches of trees. But anyways, hugely successful group or family of plants. All right, here's another example. Mistletoe. Mistletoe. Such a pain. So this is what we call a hemiparasite plant. Hemi means half. It can do photosynthesis on its own, but it just prefers not to. It's lazy. And so if it can latch on to a tree and plug in, so to speak, and siphon off nutrients from the tree, that's what it'll do. If you look at these leaves, so hemiparasitic means that hemi is, yeah, it really could do photosynthesis. It just chooses not to. And The mistletoe leaves, when you look at that picture, the mistletoe leaves are the two leaves on the right. And the two leaves on the left are the host tree leaves. You can see how complete that mimicry is. And so the mistletoe is able to blend in. They do kind of clump up. They tend to form kind of tight clumps in the branches, so you can kind of pick them out. But the leaves are remarkably similar to their host trees. So this is a red river gum tree leaves that it's mimicking. But I have down in the notes section, it says if you were to take that mistletoe and take it off the red river gum tree and put it onto another tree, a different tree, it wouldn't like change its leaf structure. It would still stay like that. It's not going to do anything different. So there's that kind of mimicry. Then there's this idea that we call heteroblastic leaves. This is more to do with the life cycle of one plant. So what you're seeing here is they look like, they're different leaf shapes, right? And so you might think this is all from different plants, but it's not. It's one plant exhibiting these different leaves over its lifespan. You typically see this as you go from juvenile phase into adult phase. So when a seed first germinates and starts growing, you often can't identify what it will be unless you know the juvenile phase of that species. So the juvenile leaves often look very different than when it becomes mature. So we say juvenile in the plant world, it's kind of like puberty in the animal world. So juvenile phase means they haven't gone through puberty yet and gone into a mature adult phase. And so the bottom leaf is the adult. That's what they're going to look like eventually, but all those top leaves are what it's doing as it's going through its juvenile phase and reaching adulthood. So we give that term, we call that process heteroblastic leaves. And then this is a nice example too of very different looking leaves. And the juvenile is this one on the left. And then it morphs into, the new leaves that come out once it becomes an adult will look like the bigger ones on the right. So that's one plant going through its lifespan, going through all of its life phases. Here's another mock-up of this idea. We're trying to show more of a progression over time, juvenile to adult. Okay, so there's that process. Then there's another idea called environmentally dependent heterophyly. This is where the leaf morphology within one plant will vary at the same time. So it's not going from juvenile to adult. It's like in the stage it's at that moment, you'll have two different kinds of leaves. Probably the most famous example of that are aquatic plants. If you've ever looked carefully at aquatic plants, when you look at especially when you have not totally submerged, so you have part of the plant above the water and part submerged, you can see that they very, very often have different leaf morphologies, depending on if they're totally submerged or if they're above the water. And then another really famous example is the carnivorous pitcher plants, where they have very special leaves for the trap, but then they have other leaves to do all the photosynthesis and that important physiology stuff. And so those are examples of, in a single point in time, they can have different leaves. So this is kind of more where we're going to be zooming in now. There's another kind of example of this that's called mimetic polymorphism. And that's really what we're going to be zooming in on now to think about vision. Mimetic polymorphism. So poly is referring to many. Morphism is different morphologies or shapes. So many shapes are being mimicked when you think about that phrase. All right, so what are we talking about? The ability of a plant to adopt the appearance of another object, such as the leaves of its host tree. so far, and I double checked just last night, I could not find any other example except for this one. There's only one plant species that can do this currently. It's called Boquilla trifoliolata. And they consider it a shape-shifting plant, which I think is a neat way to think about it. How many people have heard of Boquilla trifoliolata? Anyone? It's pretty rare. Like I said, this is the only one. Doesn't grow here, unfortunately. That'd be awesome if it did. It's down in South America. And it is a woody vine that can grow through the tropical forest. Well, they say it's a temperate forest where it's native to, a temperate forest, but really thick diversity, lots of trees, and it has the ability to grow along the ground. And then when it finds a suitable support, it'll grow up onto the host tree trunk. So this started back in 2014, where they were traipsing through this area of the forest and they came across this vine. And as they were tracking the vine, it can grow big, long vines. And as they were tracking the vine, they noticed the leaf shape was changing depending on what plant it was near. It would start to mimic the plant it was nearest to, the leaves. So this was a published study. This is what kicked it off, leaf mimicry in a climbing plant. And their hypothesis for why the plant was doing this is to camouflage itself, which is pretty common when you think about animal mimicry. That's kind of what they're doing in many cases. So this is what it looks like. And it took them a while to figure out like what is its normal shape. They had to like grow it all by itself to see what do its leaves really look like when it's just growing all by itself. And then they noticed, well, geez, depending on where it grows, the leaf morphology alters. And so what it's doing is whatever host tree it's climbing on, whatever leaves those look like, it morphs into those leaves. Its leaves will look just like the host. And down to the color, the shape, the size, the orientation, petiole length, and if there's any spininess on the tip of the leaf. Some, especially southern hemisphere plants can do that quite a bit. So this is pretty bizarre. When they came across this, they were like scratching their head. So the fancy word is phenotypically plastic, which just means that it's able to change what it looks like. And what's even the kicker is that one vine, like I said, it can be very long. And so it can be near multiple different plants. And so on one vine, you can find different looking leaves, all mimicking whatever plant it's closest to. So one plant can actually mimic one vine can mimic several different plants. Does that make sense? Okay. So when they found that out, they were flabbergasted, I think is a good word for that. And they started studying more individuals, is this a one-off or is this species capable of doing this, in total? So they really hunted around for it in that forest. And here are some pictures showing the different host species and the vine. And so I don't know if you can see can you see the white letters and arrows on there well enough? So where it says V, that's the vine, and T, that's the tree leaves. So on this top, they have the vine. So this is all the vine and then the tree. And so if you just keep scanning each figure, I mean, it's remarkable how similar the vine and the tree leaves look to each other. Here's a I blew it up here a little bit. That's pretty good, don't you think? So here is what they think it typically looks like when they were doing some more studies on it. And then they were running an experiment and showing how it can morph depending on the host tree. And so the red is the vine, the red arrow is pointing to the vine, the blue to the host tree. So this is the study that was being done in 2014. So this is a remarkable discovery. I wouldn't be surprised if they might get a huge award for this somewhere down the line, but it definitely left everyone scratching their heads going, what is going on here? How can this plant do this? And first they asked why. That's a little easier to answer. And so camouflage, as the paper title is hinting at, they say right in there, well, they're camouflaging. So if the vine, the Boquilla vine, is nutritious, more nutritious than the host tree leaves, you can imagine that Boquilla would want to hide out and camouflage, like, just ignore me, nothing to see here, just ignore me. And hopefully the host tree will get attacked and eaten and it can be left alone. And so they did look at the nutritional content of the leaves and were comparing them to the nutritional content of the host trees they were climbing on. And it was the case that very often the Boquilla vine's leaves are more nutritional. And so camouflaging makes sense. Like, I'm hiding out, I could be an easy target if I'm all by myself, but if I camouflage into the host trees, hopefully I won't get chewed on as much. So the fancy way to say that is herbivore avoidance. And when they were doing field studies, they found that very often there would be weevils on the vine or the trees or leaf beetles. And so they think that they were trying to camouflage against these types of herbivores and predators. So weevils and leaf beetles, it's important to know, they rely on vision to look for yummy things to eat, good leaves to eat, and smell. So they're not just doing smell, they're looking at what does it look like, so that they're relying on both smell and vision when they're hunting out their food, which leaves to chew on. So they started to do some research on let's look at this a little bit more carefully. So here's a table showing that if they have Boquilla growing on the host tree all by itself with no support around, or stems that don't have any leaves on it, so essentially like a pole, they could show that the Boquilla was not getting eaten as much if it was on the host tree, with the host tree having its own leaves. So the camouflage idea was upheld in their experiments because herbivory was significantly decreased if it could climb on a host tree and mimic its leaves. Now, that last bullet point was part of their study, and they didn't expect this. Mimicry was also observed when the vine was not even in physical contact with the host. So that's an added twist they did not anticipate, and kind of messed with some of their hypotheses they were formulating. I'm guessing they went, oh, shoot, when they ran across this observation. So the vine does not have to be in physical contact with its host in order to mimic. So it's important to keep in mind. So then they started going, okay, all right, what's going on here? How can this vine so carefully and completely mimic its host tree? And one vine in its lifespan can have several mimicking going on all at the same time, several different host leaves. So they're thinking, okay, it's queuing in on something to be able to do this, polymorphism. So they started to look at maybe there's something to do with light quality, like light intensity, intensity is looking at how bright or dim. Could they be somehow reading that ratio around the host leaves? Is there something going on else abiotically? So they were trying to throw out all kinds of hypotheses and ideas. Maybe there's shading going on and they're able to pick up on that and somehow morph their leaves. We're going to learn not too soon or not too far from now a little bit more about phytochromes and how plants use phytochromes in order to figure out if they're in light or if they're in dark conditions. So we'll talk about that. So they looked at phytochromes. Maybe they're using those to assess what's going on. And then, of course, hormones. They were like, oh, I bet there's hormones involved. And they haven't done so much work with that, surprisingly. I'm surprised they haven't followed that down a little bit more. But like I said, this research is kind of new. Okay, so then this idea, do you remember? So this is the dodder plant, DODDR, that parasitic plant. And here it's trying to choose between tomato and wheat. How is it choosing? Remember, we just saw this last. Yeah, smelling. It's smelling the volatiles that's being given off by the tomato. It really loves tomato. Tomato's an easy target and very nutritious. So they thought maybe Boquilla is doing something like that, like it's picking up on volatiles from the host tree, and somehow that information's allowing it to morph its leaves. And that kind of took the forefront of hypotheses, like that sounds really logical because you wouldn't need to be in physical contact then, right? So if Boquilla is not even making physical contact, that's okay because the volatiles are okay, they're going to come over and you don't need physical contact. So this was a running hypothesis for quite a while. Then another really weird one. Well, that's not so weird. The volatiles isn't really that weird. We know that plants can do this, but this is an interesting one that I still can't quite. Well, let's get into it. So horizontal gene transfer. Now, I'm a big believer in horizontal gene transfer in the sense that I see microbes doing it all the time. And so I teach general microbiology. We talk about horizontal gene transfer quite a bit in that class. And if you talk to a microbiologist, they'll say horizontal gene transfer is heavily involved in the evolutionary process and continues to be so today. So what is it? What they claim could be happening is that the host plant's DNA got transferred to the vine. So that's horizontal gene transfer. It's between two living individuals coexisting near each other. So they're saying, yep, the plant DNA, the host DNA, or epigenetic factors of the host tree is getting transferred to the vine's DNA. And then the vine expresses that DNA and it allows for it to morph its leaves. So once the vine gets access to the plant's DNA or epigenetic factors, it can start morphing its leaves. So we're talking about plant to plant transfer of gene transfer. Now, this was one thing I was hunting down last night because I did find in the literature some really good examples of this. And so they know that this can happen, but that it could happen in this case is still heavily up for debate. Let's back up and do something that they have nailed down pretty well. We know that viruses and bacteria and even fungi can do horizontal gene transfer pretty readily. And they act as vectors. So if you have learned about vectors in biology, it's usually, a vector is something that moves something from point A to point B, like the mosquito that causes malaria is the vector, for the pathogen that causes malaria. It moves around and infects people with the malaria pathogen. So what we're talking about here, this is an example of a virus acting as a vector to get DNA transferred, horizontal gene transfer. So this is with a bacteria and a virus. So you see the virus attacking the bacteria. The bacteria is a rod-shaped cell. And the virus injects its DNA and takes control of the bacteria and says, make more viral particles. And as it's doing that, every once in a while, what happens is that you get some of the host DNA packaged into the new viral particles that are getting made. I don't know if you can see where you're sitting, but the viral DNA is green and host DNA is purple. And so they're trying to show that this viral particle got some of the host DNA with it. And so now when all of these break free from that bacteria and they go to infect another new bacteria, they can inject some of that bacterial DNA from the first host. So this is the example of horizontal gene transfer. You have genes from the first bacteria getting packaged into the virus and then getting injected into the second. That makes sense? All right, so horizontal gene transfer. Well, that's a well understood phenomenon. We know that happens quite readily. We also know this happens really quite nicely. So this is the idea of Agrobacterium tumefaciens. It's a bacteria. And it can inject, so to speak, some of its plasmid DNA into plant chromosomes. Actually amazing how well it can do this. And so here you're looking at the bacteria in the purple here, and you see it has a chromosome, but we're zooming into the plasmid that it has. Okay, so that's separate from the chromosome, and some of that plasmid can get into the DNA of the plant. So this is horizontal gene transfer from a bacteria to plant. And not me personally per se, but plant biologists take advantage of this a lot in molecular biology labs because this is a really nice way to introduce a gene of interest into the plant you're interested in. And so if you put your gene of interest, maybe it has something to do with drought tolerance or disease resistance, you can put your gene into that plasmid and then mix plant cells and bacteria cells in a flask together. The bacteria will do this for you. That's really sweet how easy the bacteria can do it. Plant cells are really hard to genetically modify without the help of Agrobacterium. It's kind of challenging. So this was a huge breakthrough in molecular biology and in the plant world. So, okay, so we know that can happen. In the microbiology realm, it's really quite common. But plant transfer, that's not so common. And so here, this is a paper in 2007 where they are documenting it. But they're still like, we don't know quite sure how, how this works. And what they're zooming into is the mitochondria DNA. So remember, mitochondria has its own DNA. Ours does, plants' mitochondria do. And they say that that's the DNA that's getting transferred horizontally from plant to plant. And they found an interesting plant, Amberella. You don't need to know that, but they did zoom in on one plant that is able to do this in massive amounts, where lots of DNA is getting transferred. So they know that it can happen. And I thought this was interesting. Amberella, the leaves, the branches, the trunks, and even the fruits are often covered with diverse epiphytes. So those are other plants hanging out on Amberella. Amberella is like a short little tree or shrub. So there's mosses that hang out on the branches and other bryophytes. And they're saying that those epiphytes could be transferring their plants and they could be transferring DNA to Amberella. And so this plant physical contact with the epiphytes to the Amberella, you could get gene transfer that way too. So I thought that was an interesting thing to think about for how could this be happening. They also know about jumping genes. You might have heard that term, jumping genes, transposons. So this is dating back to when Barbara McClintock and all of her famous work with corn. Back in the 1950s, she won the Nobel Prize for this work, where she could demonstrate that chunks of DNA can get transferred from one chromosome to another in the corn genome, and that causes the different colors of kernels of corn. And amazing scientist, and just did some great research. And so in this, now they're talking about, okay, so that Barbara's work was with within the corn plant's chromosome. Now they're saying, but we can get horizontal gene transfer from plant to plant. Okay. And here it says the authors conclude that these sequences were transferred horizontally between the two plants long after they went their separate ways. And so this is with rice and millet. So some really amazing studies with the rice plant and the millet plant that's at some point back in their history, they had genes exchanged between them. So there is documentation out there. It's not huge, but there is some really interesting cases where you can see horizontal gene transfer between plant species of DNA, mostly mitochondrial DNA. So could that be going on with Boquila? Maybe. So hold that thought. Then one of the leading authors of this 2014 paper said, hey, what about not just horizontal gene transfers from, well, there's a couple things before we talk about the endophytes. The lead author on this paper, he said, what if, so this horizontal gene transfer, he's envisioning, what if there's bacteria on the host tree that waft over to the vine and do horizontal gene transfer? So that was one of his thoughts too. It's like, doesn't necessarily have to be plant DNA. What if it's bacteria that were hanging out with the host tree and somehow can epigenetically transfer this information to the Boquila vine? To me, that doesn't sound that plausible, but that was one of his ideas. And then he expanded and said, what about endophytes? Once we learned more about endophytes. And he's like, well, maybe bacteria or fungi that hang out in the host tree are getting transferred somehow and over to the vine. And that's what's causing the mimicry to occur. So we've learned about endophytes. Could the endophytes from the host tree somehow be getting transferred into the vine? If they're making physical contact, maybe, but there's cases where the vine isn't making physical contact with the host tree. I don't know. What do you think? Endophytes or bacteria wafting over? Do you buy it? I'm having a hard time with that one, even though I love endophytes. Endophytes are amazing. And the other thing, if endophytes get over or horizontal gene transfer is occurring, it would have to go systemic through the whole vining plant body, not just to one leaf, because you see a bunch of leaves changing. So yeah, I'm having a hard time with this one. Then they also propose maybe mycorrhizal connections could be causing something. Could there be some information through the mycorrhizal networks that's getting transferred? Now that is interesting, but again, there's going to be a study I'll show you where they're not sharing the same soil and they can still mimic the host. Okay. A couple of years later, after this first paper, so this is a 2016, Stefano Mancuso, who we've been listening to some of his videos and reading some of his papers, he said, hey, what about vision? Maybe the vine is seeing things. And so this is the title of his paper, Vision in Plants via Plant-Specific Ocelli. Anyone heard about ocelli? Ocelloids, if I guess is another way to talk about, well, something similar. Ocellus, I think, is singular, ocelli is plural. Although plants are sessile organisms, almost all the organs move in space and thus require plant-specific senses to find their proper place with respect to their neighbors. Here we discuss recent studies suggesting that plants are able to sense shapes and colors via plant-specific ocelli. All right. Well, that means we have to think about vision. So let's think about human vision and animal vision. So you have your eyes have the cornea on the outside to protect. Then you have the lens that's going to be doing some of the focusing. The iris, that goes open and close to further allow for better focusing. And then you have the light coming into the eye and going back to the back part of the eye, to the retina. And then, of course, you have the optic nerves that go to your brain. And within incredibly fast speeds of time, it's telling you what you're looking at. It's amazing. Let's see if we can watch this video because the video explains it a little bit better, I think, than what I can do because I am not a human phys person. Is this what I want? No. All right, let's try that again. All right.
How do we see? Let's follow the light. Light enters the cornea, the clear window of the eye. The cornea bends the light, so it passes through the pupil. The iris makes the pupil bigger or smaller, which determines how much light gets to the lens. The lens angles the light through the clear vitreous to focus it on the retina. The retina converts the light into electrical impulses. These impulses travel along the optic nerve to your brain, producing an image. While it may sound complicated, this happens in less than a second and allows you to see the world around you.
Okay, so pretty amazing what our species has evolved for an eye and vision. Okay, why don't we stop there? We need to take a break. So keep that thought. Let's get up and stretch and move around and then so it's 2:53, so like 3 minutes after 3, we'll start up again. All right, so we have the cornea, the iris that can move like a camera lens, and then the crystalline lens is helping to bring in the light and focus it back to the retina. Okay, so we have to think about this a little bit. So basically, we need some, this kind of convex idea on the surface of the eyeball and on the cornea, and then the light coming through and being able to focus it. All right. So here, Stefano Mancuso and his group were saying, could we have ocelli in plants that are capable of picking up these images? It's kind of in their own way, in their planty way. So here on figure A, you're looking at the leaf and they've taken a section of it. And so you have the leaf epidermis, the very surface. So just like us having epidermal cells, plant leaves have that too. So they have an epidermis. And then the cells in the leaf, they're showing full of chloroplasts here. So these are some of, it looks like they've drawn palisade mesophyll cells of the leaf. And of course, those have a bunch of chloroplasts in them to do photosynthesis. Okay. So what they're suggesting is down here, they're saying, okay, if you have light striking the leaf, and the cuticle is this rectangle up here, that's just a waxy covering. So they said, okay, it's going to come through the cuticle, which is like the cornea, and travel through the epidermis, which is lens-like, and then it's going to focus in, and you see these chloroplasts, but then if you look carefully, you see these little purpley pink dots. Those are the ocelli. So they're looking at the leaf and trying to imagine how could the basics of vision be working with the structure of the leaf and the leaf cells. So if you can get it focusing in through the action of the cuticle, the epidermal cells, and then getting it focused in on the ocelli. So in the notes section, I have it written out a little bit more of what they were thinking for review when you're studying. So that's in the leaf, but they're also looking at roots. And believe it or not, roots pay attention to light. They don't like to be in the light. They avoid it. They will do light avoidance because, of course, roots didn't evolve to be in light. And so if they're in light, something's wrong. And so they're trying to avoid it. So they do have to pay attention to light, even though it's kind of weird to think about it. But we're zooming in again to the transition zone. That should sound vaguely familiar from the stuff that we talked about before, how you have the transition zone of the root, a tiny section right near the root apical meristem and the root cap that's capable of, what they think is capable of kind of decision-making processes for the plant. So again, you have the epidermal cells on the root surface. Then you have the cortex cells, or just parenchyma cells below the epidermis. And again, they're showing that you could potentially get the focusing in the parenchyma cell area. Could there be something there that it's able to have some sense of vision? Okay. In that paper, they also pose this question. Isn't it curious that epidermal cells do not have chloroplasts? So epidermal cells are right on the surface of the leaf. And so the way the artist drew this, you can see these cells are empty, so to speak. I mean, they're full of nuclei and all of that, but there's no chloroplasts. And that's not an accident that they drew it that way. Epidermal cells don't have chloroplast, but they're right. Isn't it kind of weird? Because you'd think they put chloroplast right on the surface of their leaf so that they can photosynthesize the heck out of the day. You know, I can just do photosynthesis everywhere. My epidermal cells, which are most closely accessible to the sunlight, it would make sense from that perspective to dump a lot of chloroplast in your epidermal cells. So you can do a lot of photosynthesis, and yet they don't have them. So that was a piece of evidence that they're pointing to that says, maybe there's something there. Maybe these epidermal cells are being used in this manner rather than doing photosynthesis. They're a way of focusing light. And so that was an interesting proposition that was made in the paper as part of their thought process on what could be happening with plant vision. So they're saying, yeah, if you have a ton of chloroplasts in your epidermal cells, that's not going to work for the lens-like functioning that they're proposing. And so they're like, okay, maybe those epidermal cells are using the mechanism of focusing light. That's their job, rather than, they don't have any chloroplasts in there to do photosynthesis. So they published this paper, and then the original author of the 2014 paper, Ernesto Giannoli, he came back and said, no, he didn't like that. And so he published a letter to the editor that gets put into the journal for everybody to read. And he said, no, he was still favoring the idea that horizontal gene transfer, he thought, or volatiles would be the way to go. So then Mancuso's group came back to that criticism with a rebuttal and said that he doesn't like, Giannoli, the original author of the first paper, he doesn't like this idea of vision in plants. And I like how they're so polite at the very end there. It says, we think, where'd it go? Anyways, they're very polite. I read the whole thing, and he's like, we think the author for his, viewpoint, blah, blah, blah. But anyways, if vision had evolved in cyanobacteria and unicellular algae before the origin of higher organisms, which it has, I'll convince you, I hope, it is plausible that this ability has not been lost during evolution. In fact, it would be surprising to find that plants are not endowed with similar mechanisms, especially given that this feature is preserved in multicellular algae, such as Volvox. To mimic precisely complex objects such as leaves, Boquilla must be capable of sensing parameters such as shape, dimension, and color. In other words, it must be able to perceive vectorial sensory information. To do that, it would need some kind of vision. In fact, chemical components, including volatiles or RNA and DNA, cannot provide any vectorial sensory information. That's true as far as we know. Although we do not dismiss the involvement of volatiles or other chemicals, in our opinion, they only have supportive roles. Well, that was a pretty strongly worded rebuttal, polite, but making some really good points that, cyanobacteria have their own vision, way of seeing. So do algae. Volvox, if any of you've worked with Volvox in any of your labs, it's a multicellular spherical organism with thousands of cells all put together. So really nice rebuttal. So let's think about that a little bit. So this idea that there has been vision before plants, terrestrial plants came about. There's been vision in organisms. So they point to the cyanobacteria. Yes. So this is a paper that was published back in 2015 that the entire cell of the cyanobacteria can act kind of like an eyeball and focus light and pay attention to light and either go toward it or away from it. So if you're a cyanobacterium, you generally want to go toward light because you can photosynthesize. That's one great feature of cyanobacteria. So they found this in a particular species, Synechocystis species. And so that one has been heavily studied. And they are looking at, like I said, like the whole cell acting like an eyeball, so to speak, where you have the focusing of the light to a point like that, similar to how it does on the back of our retina. And then it can interpret that and either move toward or away from the light. Let me see if I can get this to play. There's a little video showing. Okay. So this is from their study. I don't think there's any words to this. It's just, so they're spotted onto that, and then they dry and put a cover slip on it, and then they seal it with silicone. And then they do unidirectional light from a couple of different directions. And then hopefully they're gonna play the movie. Okay. So here, the single-celled cyanobacteria. Now they've enhanced it with color, and then they're going to do the tracing of how the path of movement they'll show with tracings. So now you can see the light is coming from the right. You can see the shifting toward the right, toward the light. And if you see the tracings, now you can really tell that they're moving toward the light. And then the top is lit, and you can see them moving up toward that light. And then they'll color code it again to make it easier, and then they'll do the tracing. So clearly they're sensing the light. They're able to detect the light. I mean, it's not just one or two in there random, that it can just be attributed to some random movement. This is mass movement of most of the cells toward the light. And here they did both lights to see what would happen, and you can see them kind of shifting, they're picking which one to go to, whatever one is closer. So that's in the notes section. There's the link for that in the notes section to that slide if you want to watch it again. Let me see if I can. All right. So nice study showing that cyanobacteria are doing this. So remember now, bacteria were here 3.5 billion years ago. They were the first entity. Cyanobacteria came after the first prokaryotes because cyanobacteria are a little special that they can do photosynthesis. So they came a little bit after 3.5 billion years ago, but not too much after that. And so we're talking, the first life forms are able to do this. And it's not something new. Like that paper was published in 2017, but they point out this is not really, we've known about this for over a century now. So synechocystis cells act as spherical microlenses, allowing the cell to see light source and move towards it. Spherical cyanobacteria, are probably the world's smallest and oldest examples of a camera eye. So that is pretty convincing evidence. Then they show this idea of Chlamydomonas, which is not a cyanobacteria, that's a green algae. So now we're jumping into the world of eukaryotic organisms. It's an algae. And Chlamydomonas reinhardtii is like the guinea pig of green algae species. And they have known for a long time that it has an eye spot, and that has been reported on for a long time. So here is a paper that was published in 2022 where they're re-examining this idea in conjunction in supporting evidence of, higher plants could have this ability too. So there's Chlamydomonas hanging out, single-celled algae, and you can see its funky little eye spot, and then they've blown it up here. You don't really need to know the details of its structure, just that it does have an eye spot and a way to focus light and pay attention to vision, see vision, see the light, so to speak. And so eye spot, in the paper, they're calling it oceloids. They're using that word, basically the same thing. Okay, so we're up into the eukaryotic world. Let's continue on with that. So dinoflagellates are in the protozoa, protists, kingdom Protista, so eukaryotic, and they have oceloids. These are funky. They're built from endosymbiotically acquired components. So this is really bizarre. This is the idea that you have here dinoflagellates, some of which have an eye-like oceloid consisting of subcellular analogs to a cornea, lens, iris, and retina. Here we show using a combination of electron microscopy, tomography, isolated organelle genomics, single-cell genomics, that oceloids are built from pre-existing organelles, including a cornea, geez, I'm having a hard time talking today, cornea-like layer made of mitochondria and a retinal body made of plastids. How bizarre is that? We find that the retinal body forms the central core through an ancient endosymbiosis with a red algae. So here you have this endosymbiosis event that combined the mitochondria and the plastids, put it all together, and you have an oceloid for this type of organism. Bizarre. And so here is their image of it. And again, you don't really need to know all the anatomy, but basically that they are, finding this camera-like structure. This one was made from different organelles. That's just bizarre. Cornea-like mitochondria, so that's in the blue here. And then the plastids in the back for like the retina. So that's just super bizarre. And so this is through endosymbiosis that they think this has occurred. Just bizarre. All right. So also in this publication, they went into the root zone, and we're looking at that more carefully. And they know that roots try to get away from light. So here you have a Petri plate, half lit and half in the dark, shaded. And they put the little seeds on the lit side, let them germinate, and you can see the radical root coming out of the seed and the little seedling. And you can see how they're all curving toward the shaded side of the Petri plate. And so in this case, they're avoiding the light, they're detecting the light and avoiding the light. And they think it's happening in the transition zone where you have the retina-like, that blue area that they're showing here in the back of the cells in the root is like a pigment, they're saying it could, and is paying attention to that light striking it. And then they're using that information to grow toward the dark, grow away from the light and get into the dark. So the PHOT1, they have it there. PHOT1, that's a photoreceptor. We'll talk more about photoreceptors in the near future here, but they're saying that that could all be playing a role in detecting the light. And in this case, the roots are bending away, going away from it and trying to find darkness. So the fancy word for that is scototropism. If an organism is trying to get away from light, a plant's trying to get away, like roots do, then they call it scototropism. So they're saying, yeah, the roots have to see in order to do that. They have to detect that light. So they know that when you get into even more sophisticated, complex multicellular organisms in the eukaryotic world, like cephalopods and cuttlefish, they see over their body surface. It's not just in one spot like we do with our eyes. Sea urchins and brittle stars, they also have a dispersed visual system. And there's other animals that have what look like ocelli. It's just the example of starfish. They have compound eyes at their arm tips. The medusa have eyes at the bases of their tentacles. So they're coming up with a lot of other organisms. In conclusion, it emerges that vision is an ancient sensory faculty which evolved some 3 billion years ago with the very first cyanobacteria. Evolution never discards successful innovations, and the algal and plant vision is based on that of chloroplasts too. So they're making a very strong case that, hey, this has been around since bacteria were here, cyanobacteria in particular. So they're claiming that the plant can see over its whole body with what's going on in the leaves, focusing the light in that regard, the roots, with the transition zone of every single root. And so they're claiming that the Boquila vine has that type of vision over its body where it can interpret the host tree's leaf morphology and then dynamically shift its morphology. What do you think? Vision in vines? Well, right before that one was published, this got published. And it's the same author who first discovered Boquila to begin with. He's still dead set on bacteria transferring. So this was a paper published in 2021. Endophytic bacterial communities are associated with leaf mimicry in the vine Boquila trifoliolata. So Ernesto Giannoli is the first author on the original paper. Okay, so now he's saying endophytes. That could be a good option. So what they did, they looked at gene sequencing. So they were doing DNA sequencing and they were looking at endophyte communities in the host tree, the Boquila that was mimicking the leaves of the Boquila that was showing mimicking behavior, and then Boquila leaves that were not mimicking. And they said we found significant differences in the endophytic communities. The mimicked Boquila and the host tree showed an overlap concerning their communities. The role of bacteria in this unique case of leaf mimicry should be studied further. So there's an overlap. So he's saying there's similar endophytes were showing up in the host tree plus the Boquila that was wrapped around that tree. The leaves were both showing. Now, yeah, they could have similar endophytes because they're close to each other, hanging out together. I could see that. And so, yeah, maybe they are playing some kind of role. However, another study was done in 2022 that was really interesting, but it was a small study. Boquila trifoliolata mimics leaves of an artificial plastic host plant. So now they went with totally artificial. So there's no endophytes in that plastic plant. There's no volatiles. So what happens? And so they did this experiment. Unfortunately, the sample size was very small. I think they only used 4 plants in this study. So definitely they need to ramp this up and do a bigger experiment. But they had an artificial vine in the presence of the Boquila. It was observed that mimic leaves showed altered leaf areas, perimeters, lengths, and widths compared to non-mimic leaves. We have observed differences in the leaf veination patterns as well. Okay, so they were collecting data, looking at on the left, that's the non-mimic leaf, and on the right is the mimic leaf. So you can see that compared, so essentially A is the control, and you can see that there's definitely a shift in morphology. The way they set up the experiment was to have the plants in pots, and then they had light, like a barrier, a solid barrier, shelf 1 and shelf 2. Those are solid barriers. So the control leaves are down on the bottom. Okay, non-mimic leaves are down at the bottom of the same plant. And then can you tell there's this red vine that they wrapped above the shelf? And then those leaves, they looked to see what was going to happen to those leaves in the presence of the plastic vine. And they could show that those leaves were starting to shift their morphology compared to the ones that didn't have the vine near it in the same plant. So those plastic leaves don't have endophytes. They don't have volatiles. They don't have DNA. So this kind of, not kind of, this does support the idea that plants have vision, their own version of vision. And if it stretches all the way back to cyanobacteria and evolution doesn't give up on a good thing, it's plausible. What do you think? Plausible? Thumbs up? Thumbs down? It's plausible, I think. But this is still meeting with a lot of argument. This is really, it's kind of funny how much it's really ruffled some feathers and is causing a lot of argument and debate. So sometimes very polite and sometimes not so much. This is a really cool image. Look at that. Those 3 things in the center, those are ocelli. And I don't know if you can make sense of it. This is a yellow jacket. And there, over here, the eyes, you don't see the full eyes. You just see. But that's an awesome picture. That's taken from a competition of magnified photos. They have competitions every year for those. So yellow jackets have ocelloids too, or ocelli, I should say. So, okay, so this is the session on plant vision. So we can think about that and you can decide what you want to do on that one, how you want to go. But let me talk to you before you leave. I need to talk to you about the assignment. So, and also I will be posting, I've already posted some videos and readings in EDUCAT for this next exam, supporting material we're covering together in class. I'll be posting several more on vision. And then in EDUCAT, there's a section on assignments for this semester, fall 25. And I wanted to go over, I posted a PowerPoint in that to describe the assignment. Let me get to that. So this first assignment is setting you up because we're going to be doing group assignments soon. So I sure hope attendance improves because you're going to have to depend on your groups to cut the workload. But this first one will be independent. So this is a way for you, the objective is for you to learn how to read, critically read, assess, and summarize a body of work. And I want you to be able to then relate it back to what we're learning in class. We're going to do this through PowerPoint. You're going to create a PowerPoint slide set. And I'd like you to focus on what we've been covering over the first many weeks of the semester here, something that we've been talking about, you know, plant senses. If you want to do the neurobiology idea, that's fine too. Ideally within the last 5 years of publication. And what you should do is email it to me, the paper. And please put in the subject line of your email, BI 495 paper 1. Otherwise it's going to get lost in my inbox. I have thousands of emails in my inbox. So, and I get so many per day. It's ridiculous. So please do that so I can easily find it. And then wait for me to respond to your email and give you the okay. Because some papers you might pick are pretty gnarly and I might like, maybe not that one. Or whatever. So please wait. And then this is your assignment to develop a PowerPoint slide set. It doesn't have to be huge. Basically, you want to pretend like you're going to have to teach somebody about this particular paper and what was covered. So you want to give a little bit of intro. Why is it important? Why should we care? The very basics of the experiment. You don't have to be a master at what they did in their methods. Okay, I'm not asking for that. Just the basic idea. And then the key results and conclusions. And then you should also at the very end here, it says, how does this paper compare to what we've been learning in class on that particular topic you chose? So I need you to reflect a little bit. It's kind of like reflecting on, does this paper support what we talked about, refute what we talked about? How does it fit in? And so please do that. It's due on October 10th by 10 p.m., 10:10. All right. All right. And so if you have any questions about this as you're working through it, let me know. But you're going to be doing something similar to this as groups eventually. And then we're going to start like doing a lottery system of who's going to present out to the class. It's going to be very chill. You're just going to sit there and I'll post it up and we'll chat about it. Okay, so it's nothing major. It's not like you have to give it this big long speech, but I want to start doing some group work where we can start talking out as a class about some of these articles. So some of the articles, if I did not cover the article in detail in the PowerPoint slides, it's fair game. So you'll see that some things that I've posted already, I didn't really talk about in huge detail. Like I didn't pull figures and put it into PowerPoint. So if you want to grab some of those, that's fine too. You don't have to find one I haven't talked about or mentioned or uploaded. So just let me know, send it via email. Questions? All right, go enjoy that fine weather out there if you can.